Those two types of synapses we mentioned, there's an electrical and there's a chemical. And so the electrical, all that we'll say about it is on the next slide here. Okay, so basically the electrical synapse takes place through gap junctions. Now we're going to talk a lot about what gap junctions are. We're going to see them a lot through the other systems. And they're basically a channel or pore or passageway that facilitates the communication between two cells. All right, so this is a gap junction. And this is inserted in the membrane of both cells. And this is how they communicate with one another. And so several of these proteins called connexons, they're like little Legos that stack themselves up to form this passageway, and they facilitate the bidirectional movement of these electrical impulses from one cell to its neighboring cell. And so electrical synapses are rapid in communication, much faster than chemical synapses. And that makes sense because we're using electrical signals here. With chemical synapses, we actually use a different mechanism. We use vesicles and neurotransmitters, and that takes time. We're going to see why that takes time. But understand that an electrical synapse is going to be much faster than a chemical synapse. It's usually bidirectional, so there'll be bidirectional flow here of electrical impulses. So think about action potentials being fired or being communicated from one cell to the other and then in the opposite direction, okay? Whereas we know that chemical synapses can only travel in one direction, so they're unidirectional. We see excitation and inhibition happening across, so we can have excitatory signals being sent, we can have inhibitory signals being sent, and it's all happening through those gap junctions. And this is not very often found in the body again, okay? So much like I mentioned earlier, we're not going to talk about this a whole lot. It's usually found in these rare areas, the retina, the cortex, the brain stem, and other parts of the pons and so on. And so it's not very well understood scientifically either. In fact, historically, scientists thought that electrical synapses were only found in the developing brain and not at all in the adult brain. And we come to realize that that's not true. We do have them in some of these rare areas, and they're not very well understood. The electrical synapse, on the other hand, much more common throughout the body, much more well understood and described in terms of the exact sequence of events. And so we talked about this whole anatomy of the connection. It's a presynaptic with a postsynaptic, and there's a cleft in between, right? So that area where the two neurons meet, they don't meet flush against each other. There's actually a gap there. And that gap or that space we call the synaptic cleft. It's about 30 to 50 nanometers wide, so not very wide, and it just facilitates the movement of that neurotransmitter across the gap. We'll talk more about where the neurotransmitters come in. So this is usually axodendritic, as we mentioned. So this type of connection, axon to a dendrite, whereas some can be axosomatic, axoaxonic, and so on, and the different variations in the connections are shown here. There's also dendrodendritic, very, very, very rare. I think this is only found in the thalamus and other very intricate neural connections, so not at all very common. Okay, so take-home message here, different flavors of connections. The one we're really going to focus on are chemical, and the chemicals that we're really going to focus on are axodendritic. Okay, so underline that in your notes, highlight that, share a highlighter. All right, moving on to the anatomy of the synapse. So what is taking place when we really zoom in to that connection? What's happening on that first cell or first neuron? And what's happening on this cleft? What's happening on the membrane of the second cell? So we're going to go through all the events that take place from this neuron to this neuron. But before we do so, let's sort of explain what's happening here. Let's describe some terminology and some of the players that are involved. So the big picture here, this is our first neuron, this is the second neuron, and we've spoken a lot about the action potential. We talked about it, graded potentials being observed here, them being summed at the axon hillock, lots of voltage-gated sodium channels along the axon, right, facilitating the propagation of that action potential. But when it gets to the axon terminal, there aren't voltage-gated sodium channels at the axon terminal. There are voltage-gated calcium channels. And so when we get to about this ending, where this actually swells into that axon terminal, the Bouton area, the channels on the membrane changes. So these are voltage-gated calcium channels, and these are going to be really important in the transmission of this signal. So the presynaptic axon terminal is where this is starting off. This is where our vesicles are. We have neurotransmitters stored in these vesicles. So inside this vesicle here, we can see whatever X neurotransmitter. We're going to talk about what they are, what some examples are. But for right now, we're just really sort of highlighting the big picture of the process. So don't be too bothered about what the neurotransmitter is exactly. The voltage-gated calcium channels, as I mentioned, are lining the membrane in this region and are going to be very important in facilitating the transmission. The synaptic cleft is the space here, okay? Very small, very narrow space, which facilitates the movement of those neurotransmitters. And then we have a few structures here on the membrane. On the postsynaptic membrane, we have receptors and we have enzymes. So the receptor is going to be modulating the response in the second cell, and it's going to be unique to whatever this neurotransmitter is. So let's say neurotransmitter X, this is going to be receptor X. It's going to uniquely bind neurotransmitter X, right? So very specific binding happening. And then we also have an enzyme. Now this enzyme, we're going to talk more in detail about what the name of it is, but as for right now, I want you to understand that this enzyme is responsible for breaking down any excess neurotransmitter that's left lingering here in the synaptic cleft, okay? So the receptor should bind the neurotransmitter, but in actuality, there ends up being excess left around after the signal has been transmitted, and that's the job of this enzyme, to sort of gobble up the excess, like a Pac-Man type, the excess neurotransmitter that's in the cleft. We also have reuptake molecules. So there's another option for those excess neurotransmitters. We can simply reuptake them into the axon terminal. We can recycle them, repackage them into a new vesicle, and instead of wasting some of that neurotransmitter, we can repurpose it, okay? So those are some options for what happens to the excess neurotransmitter at the cleft.